everyone, welcome to 10-Minute Physiology. In today's video, I'm going to give you an explanation of how oxygen is transported from the blood into the interstitium, which contains both the cells and the fluid surrounding those cells. So with that, let's give it a go. So I'd like to start off by first discussing how oxygen is transported. Now, in order to understand that, we have to remember the components of a cell. So this is a picture of a cell, and we're going to zoom in at the plasma membrane of the cell. So remember, the plasma membrane contains both proteins as well as a lipid bilayer. Now, the thing to remember about this lipid bilayer is that it's hydrophobic in the middle. So these tails, these lipid tails, are hydrophobic. And hydrophobic things like to be with nonpolar things. And oxygen is a nonpolar molecule. So what happens is since oxygen is nonpolar and it's a small molecule, what happens is that oxygen will just simply pass through the plasma membrane. It can pass through without the aid of transporters because it's nonpolar. So let's take a further look at how oxygen is transported in the context of a capillary. So this right here is an endothelial cell of the capillary wall, and inside the blood we have a higher oxygen concentration than outside the blood. So because of this, what happens is that oxygen will flow down its concentration gradient from the blood into the ISF via diffusion. So in other words, oxygen flows down its concentration gradient by diffusion, and it flows from an area of high oxygen levels, which is the blood, to an area of low oxygen levels, which is the interstitial fluid. So now that we know how oxygen itself is transported, let's take a look at how oxygen levels vary at different distances from the capillary center as well as at different lengths of capillaries. So this right here is a figure showing a capillary, and the capillary, as we know, is going to be made up of endothelial cells. And inside the capillary we have blood, which is full of oxygen. Now what we're going to be doing is we're going to be looking at the oxygen levels at varying distances away from the capillary. So this ruler is simply showing you different distances away from the capillary. So the further down the ruler, the farther away we are from the capillary. So what's going to happen is that oxygen is going to flow down its concentration gradient from the blood into the interstitial fluid. Now, along the way, we're going to be losing oxygen. And in order to understand that, we will look at a specific number, 10 oxygen molecules, and we're going to start off at this point. So we start off with 10 oxygen molecules leaving the blood. So at this distance away from the capillary, we're going to lose around 4 oxygen molecules. And that's because the cells in this area of around the capillary are going to be metabolically active. Therefore, they're going to be drawing oxygen away from the capillary. Now, as those oxygen molecules move down, we're going to be losing more and more of those oxygen molecules. So here we're going to lose 3 more. And then if we move further down, we're going to lose 2 more. And then if we move further down, we're going to lose 1 more, to a point where we have no oxygen molecules left out of those 10 oxygens. So the main point that I want to give you here is that as we increase the distance away from the capillary, we decrease the amount of oxygen. So now we're going to look at how the oxygen level varies down the length of a single capillary. Now, along the length of a single capillary, you have all these cells, and these cells are going to be metabolically active, so therefore they're going to be drawing oxygen from the blood. So in this case, in this example, we're going to start off with 20 oxygen molecules. Now, as we move down the blood, these 20 oxygen molecules are going to be depleted. So in these cells, each of these cells will maybe take 5 oxygen molecules. So now we only have 10 oxygen molecules left in the blood. And as we move further down, each of these cells is maybe going to take 3 oxygens. And we're only going to have, in this case, 4 oxygen molecules left in the blood. And then the remaining 2 cells are going to take what's left. So what we see here is that as we go down the length of a single capillary, the oxygen level inside the blood decreases. So now we're going to look at how oxygen is used at the macroscopic level, because when we're talking about oxygen usage, we're typically talking about it at the level of organs. So this right here is a simple diagram, and it's showing you the artery that's feeding a particular organ, the organ itself, as well as the vein that takes all the deoxygenated blood away. Now, in order to understand oxygen usage, we're going to look at the different levels of oxygen of the blood flowing into the organ and the blood flowing out. So the blood flowing into the organ has an oxygen level of 20 molecules, and the blood flowing out has an oxygen level of 12 oxygen molecules. Now, you can see here that the organ, if you take the difference between the oxygen level in the artery and the oxygen levels in the vein, has a arteriovenous difference of 8 oxygen molecules. So the arteriovenous difference is simply how much oxygen is used by a particular organ. So it's the difference between the oxygen levels in the artery feeding an organ and the oxygen levels in a vein leaving the organ. So the arteriovenous difference, which we see here, can actually be equated via Fick's principle. So this right here is Fick's principle, and what it's doing is it's relating metabolic rate, which is symbolized by Q, F, which is flow or cardiac output, and the arteriovenous difference together. So what does this principle tell us? So the first thing that it tells us is that as the metabolic rate of an organ increases, the arteriovenous difference increases. So basically what happens is if you have a more metabolically active organ, it will draw more oxygen out of the blood, so therefore the blood leaving the organ is going to be more deoxygenated, therefore increasing that difference. The other thing that we see with Fick's principle is how flow relates to the arteriovenous difference. So when flow increases, this means that the blood inside the vessels is going to be moving faster. Now, whenever you move blood faster, this actually decreases the ability of oxygen to diffuse across the capillary walls. So therefore, the arteriovenous difference is going to decrease. So this relationship is going to allow you to understand how the organ uses oxygen in the context of its metabolic rate, as well as the context of the flow that's entering into the organ. So in summary, we saw that oxygen can move across cells via simple diffusion, and it moves down its concentration gradient. We also saw that as we increase the distance away from a single capillary center, the oxygen level decreases, and as we move along the length of a single capillary, the oxygen level decreases. And lastly, we saw how Fick's principle is a good way to look at the oxygen consumption at the organ level. So I hope this video helped you understand a little bit about how oxygen is transported and how it's used, and I hope to see you in the next one. See you next time.